Good morning, dear student. After completion of the concept of antigen, today we will discuss about the structure and functions of immunoglobulin. After completion of this topic, we will able to know the definition of immunoglobulin, their general structure, several varieties or types, the structural variation according to their types, functions of immunoglobulins and their clinical significance. So let's start with the definition. So what are immunoglobulins? Immunoglobulins are high molecular weight glycoprotein molecules that are produced by plasma cells in response to an immunogen and function as antibodies. So mainly 5 types, that is GAMDE, and 5 characteristics. Number 1, high molecular weight. Number 2, glycoprotein. Number 3, produced by plasma cell. Number 4, in response to immunogen. And number 5, function as antibodies. Now, why they are called immunoglobulin? Okay, it has been observed that when an antibody containing serum is placed in an electric field and electrophoresis, then the immunoglobulins or these type of proteins take the or run in the with the globulin molecule. That means it takes the portion of the globulin molecules. So according to their electrophoretic mobility, they call the globulin which are related with the immunity, so they are called the immunoglobulins. So immunoglobulins are mainly the term which is related to their physical properties. Now, what are the general characteristics? We have all come to know in the previous two slides that they are heavy protein, they are globular protein, their glycoprotein belongs to gamma globulin, okay, produced by plasma cell and in response to immunogen. And two extra points are, that is, they constitute 25 to 30% of the total serum proteins and present in the body secretion, in serum, in tissue fluids, in mucosal surfaces, etc. Okay, so these are the general characteristics of glycoprotein or immunoglobulin. Now let us see the structure of immunoglobulin. So we can see from this picture that immunoglobulin is Y-shaped structure. It composed of 2 types of chain, that is one light chain and another heavy chain. Light chain and heavy chains. So it has got 2 heavy chains and 2 light chains. So it is heterodimer. It is tetramer, but it is heterodimer, just like hemoglobin. Okay. Now, like any polypeptide chain, it also has 2 terminals, that is one N terminal or amino terminal and another carboxy terminal or C terminal. It is divided into 2 regions, variable region and constant region. Okay. So both the heavy and light chain has got single variable region, but the number of constant region varies in light chain from the heavy chain. The light chain has got single constant region. So single variable and single constant region in case of light chain. But in case of heavy chain, it has got 3 constant region, that is CH1, CH2 and CH3. So 1 variable but 3 constant region in case of heavy chain. There are several disulfide bonds present in the immunoglobulin structure. It may be intrachain, that means within a single chain, or interchain, that means between 2 different chains. So interchain disulfide bonds are present between light chain and heavy chain and between 2 heavy chain in the stem or stalk portion, but not between light chain in the 2 sides. So there is no interchain disulfide bonds exist between 2 light chains. So between light chain and heavy chain and between 2 heavy chains. Okay. There is also hinge region. Hinge means joint, just like our body joints. Joint region or hinge region, it is present between CH1, that means constant region 1 of the heavy chain, and constant region 2 of the heavy chain. So hinge region between CH1 and CH2. And there are also some carbohydrate molecules attached with the immunoglobulin. Most commonly it attached in the region of CH2, but it can be present in anywhere. So there are carbohydrate molecules in the immunoglobulin structure. That is why they are called glycoprotein molecule. Now, according to the function, it can be divided into two portions, FAB and FC. FAB means fraction for antigen binding. Antigen binding site. So, there are two antigen binding sites. So, the immunoglobulin molecule is bivalent. So, each immunoglobulin molecule can bind with the two antigen. It has got complementary sites for two antigens. So, FAB for antigen binding and FC for fraction crystallizable. That means which fraction can be crystallized. Now, we will see the structure in detail. Remember that you have to practice drawing this structure of immunoglobulin very seriously, because whenever the question comes from the immunoglobulin, we have to draw, and proper labeling is very much essential for structure of immunoglobulin. Okay, so practice it. Now, if you study it in detail, then we can see, we have already seen that it has got 4 chains. So they are tetramer, 4 polypeptide chains, 2 heavy and 2 light chains. These light chains are similar in all immunoglobulins. Okay, so light chains can occur in 2 varieties. One is kappa, another is lambda. So either kappa or lambda can be present in any immunoglobulin molecule. Okay, but the heavy chains are structurally and antigenically distinct for each classes, and the naming of the immunoglobulin also depends on the type of heavy chain present in that particular immunoglobulin. There are 5 types of heavy chain present, that is gamma, alpha, mu, delta and epsilon, according to which there are 5 broad classes of immunoglobulin, G, A, M, D, E respectively. Okay, so 2 heavy chain and 2 light chain. Light chains are identical, kappa, lambda, and heavy chains are 5 types. Okay, now there are intrachain and interchain disulfide bonds. We have already seen that. So these interchain disulfide bonds, this number varies among different immunoglobulin molecules, and these chains also binds with the help of non-covalent interaction also. In case of intrachain disulfide bonds, intrachain means within a single chain. Within each polypeptide chain, this intrachain disulfide bonds helps to form the loop, and this loop is completely folded to form a globular structure called domain. You all know what is domain, isn't it? So the intrachain disulfide bonds forms loops which compactly folded to form a globular structure called domain. Now there are variable and constant region, okay, in the, or we call variable or constant domains. Okay, both the heavy and a light chain can be divided into 2 regions, and these variable, what is the source of the name? That is variable region. That is, the amino acid sequences can be vary in these variable regions. So there are various variability in the amino acid sequences in the variable region, whereas in case of constant region the sequence almost remains same. So variable region, which is present towards the N-terminal end in both the chains, that is light chain and heavy chain, and constant region faced towards the carboxy chain, where the amino acid sequences remain almost same. Light chain has got 110 amino acids each for variable as well as the constant region, whereas in the heavy chain, variable region composed of 110 amino acids, but the constant region, as there are 3 constant regions, so there are almost 330 to 440 amino acids present in the constant region. Now, hinge region. Hinge region, we have already seen that hinge region present between the CH1 and CH2 of the heavy chain. Okay, and this hinge region, just like our joints, body joints, we can move the body parts in the joint portion, isn't it? So here also some flexibility can be possible in the hinge region or the joints region. Now, oligosaccharides means carbohydrates. These carbohydrates molecules are attached with the heavy chain, and most commonly it present or attached in the CH2 region of the immunoglobulin, but it can be present anywhere. Okay. Now, this light chain and heavy chain gene, in case of human, they are restricted to particular gene loci. For heavy chain, this is present on the chromosome 14, whereas kappa light chain present in the chromosome 2 and lambda light chain present locus present in the chromosome 22. So for the HC locus, it is chromosome 14. For kappa LC locus, it is chromosome 2, and gamma LC locus, it is chromosome 20. Now, immunoglobulin can be produced by multiple genes. It has been observed that light chain, it is produced by 3 separate genes. Namely, you can tell 2 at least, that is variable region gene, constant region gene, and also joining region gene. So V gene, C gene and J gene. So light chain is produced by 3 separate genes: V gene for variable region gene, constant region gene and joining region gene, and heavy chains produced by 4 separate genes. 4 separate genes, namely variable region genes, constant region genes, joining region genes, and one more diversity region gene or D gene. So V, C, J and D for heavy chain. Okay. These multiple genes are responsible for synthesis of a single immunoglobulin molecules. Now, what is antibody diversity and how it occurs? Okay, it has been observed that a person is capable of producing antibody to almost an unlimited range of antigen. Uncountable range of antigen can be defended by the production of antibody. Humans have multiple genes for production of antibody, but not millions of genes separately to code for separate these millions of type of antibody which can produced against unlimited number of antigens. So how these type of huge range of diversity is possible for antibody, that is several different kinds of antibody against uncountable numbers of antigen, how it can be possible? This antibody diversity is achieved by specially 2 processes, namely the combination of various structural genes, that means the recombination of various structural genes. We have already seen that the variable region, there are the amino acid sequence differ in variable region. So it is also produced some diversity because amino acid sequence, the several sequence can be occurs in the variable region, in light chain, in single light chain, in both light chain, in heavy chain, in both heavy and light chain. So several types of sequence can occur in permutation and combination, produce several millions type of antibody. Other than this, the recombination during production of immunoglobulin, recombination can occurs between the genes. Okay. So we can't discuss it in elaborated in here because you are too small for that. And the second mechanism is somatic mutation or somatic hypermutation. You just remember the recombinations and somatic hypermutation produce the antibody diversity. To know the function properly, the researcher cleave the immunoglobulin molecule with the proteolytic enzyme. We all know it is glycoprotein. So proteolytic molecules cleave the immunoglobulin molecules. Proteolytic enzyme cleaves the immunoglobulin molecules so that we can study it in more detail. So if we digest the immunoglobulin with the papain molecule, papain, we all know papain is an enzyme which present in the papaya. Okay, so papain enzyme, it cleaves the bonds before the hinge region. That means before the HH interchain disulfide linkage. Okay, so here if the break occurred before it, so how many fragments will produce? It produce 1, 2, 3 fragments. So 2 FAB fragments, that means fraction for antibody binding, antigen binding, and on another FC region or FC for fraction crystallizable. It has been seen that this lower portion can be crystallized. Okay, so the FAB, it contains the variable region of both the heavy and light chain and constant region of the light chain and the CH1 of the heavy chain, whereas the FC portion composed of CH2 and CH3 of the heavy chain. Okay, so there are 3 fragments, 2 FAB and 1 FC. Now we have already discussed this in your previous slide, that is there are FAB and FC2 FAB portion and FC portion, and FAB portion composed of variable region of both the chain and constant region 1 of the heavy chain and constant region of the light chain, and CH2 and CH3 domains of the heavy chain falls under the FC fragment or fraction crystallizable. Now if we digest the immunoglobulin with pepsin rather than pepsin, so where it will cleave? It will cleave the heavy chain after HH interchain disulfide bond. So papain cleaves before HH bond disulfide bonds, heavy chain to disulfide bond between 2 heavy chains, and pepsin cleaves the bonds after the interchain disulfide bonds. So if it cleaves here, so these fragments remain joined, isn't it? So it will produce FAB2, that means as a whole FAB2, not 2 FAB. So not 2 FAB, FAB2. See the difference. FAB2 means we all know, we have come to know, that is the immunoglobulin molecule is divalent, so each the arm can bind with one antigen. In case of papain digestion, it can bind the antigen but separately, but here it can bind 2 antigen. That means the valency 2 remains constant. So here it is called FAB2. Produces single fragments composed of 2 FAB-like subunits called FAB2. Okay, because it is divalent and can bind 2 antigen at a time. Okay, and FC fragments, in this case FC fragments are not recovered because it has been digested to numerous small peptides. So FC fragments cannot be recovered like papain digestion. Okay, here the comparison of 2, very easy, that is papain cleave here, so produce 2 FAB, each is monovalent, and FC, whereas pepsin digestion produce FAB2 which is divalent, and this FC portion cleaved into several numerous peptide, so cannot be reconstituted or crystallized. Okay, now next the classification. So we have already know that the naming of the immunoglobulin based on the type of heavy chain present in that particular immunoglobulin, and they are gamma, alpha, mu, delta and epsilon. So the immunoglobulins are G for gamma, A for alpha, M for mu, D for delta, and E for epsilon. Now comes to individual immunoglobulin one by one. First starts with the immunoglobulin G or IgG. So this IgG is most abundant in our serum and comprise of 80% of total immunoglobulin. They present in almost everywhere, that is blood, plasma, tissue fluids, etc. And it is called 7S immunoglobulins. S for Widberg unit, sedimentation coefficient. Okay, so it is 7S. Okay, different immunoglobulin has got different sedimentation coefficient. Okay, now it contains less carbohydrate than other immunoglobulins, and it has got longest half-life, that is 23 days. It is the longest of all immunoglobulin isotypes. So what are the most thing that is most abundant, that is 80% in the serum. Number 2, longest half-life, okay, 23 days, and less carbohydrate than other immunoglobulin molecules. Now what are the function? This immunoglobulin IgG, being small molecule and monomer, it present in as single molecule. So monomer, so being small molecule, it can cross placenta, that means from maternal to fetal circulation, and provide immunity and give immunity to the neonates at birth. Okay, it acts against bacteria and viruses by opsonizing and neutralize the toxin. Opsonization means the engulfment or engulfment of the bacteria and neutralize their toxin. It activates complements by classical pathway. There are three types of pathway for complement activation. So it activates the complement by classical pathway. It's secreted in high quantity in secondary exposure. What do you mean by secondary exposure? That means whenever we first expose to a toxin, first exposure with a new toxin, then antibody produced to defend it, and this is mainly IgM type of antibody, and some antibody remains as produced memory cell. So next time or second time when we face the same antigen, okay, we recall our first encounter. Yes, we have met before, so we have to fight it back quickly. So this is our enemy, so we have to fight it back immediately, because we can identify the enemy, because it is our second exposure or the subsequent exposure. Okay, so this is the secondary exposure. The secondary exposure, the main type of immunoglobulin is IgG. The catabolism of IgG is unique in that sense, that is, it varies with its serum concentration. Now, what are the subclasses of immunoglobulin G? There are 4 subclasses, that is immunoglobulin G1, 2, 3, 4. How they vary from each other? Mainly by the number of the disulfide bond. Number of disulfide bond and the position of disulfide bond. Number and position of the disulfide bonds. Okay. The most number of disulfide bond, interchain disulfide bond present in IgG3, okay, followed by IgG2, where 4 disulfide bonds are present in the hinge region, whereas both in Ig1 and Ig4 only 2 interchain disulfide bond present in the hinge region. Okay, so how they differ? That is, the Ig1 differ from Ig4. They differ from the position of the disulfide bond, interchain disulfide bond between the light chain and heavy chain. See the position here. The position is toward the carboxy terminal end of the light chain. Okay, in case of Ig1, but in case of Ig4, the position of the interchain disulfide bond between light chain and heavy chain varies. Okay, so the subclasses of IgG, that is Ig1, 2, 3, 4, they varies according to the number and position of the disulfide bond. Most of the disulfide bond present in the IgG3. Okay. Now, what is the biological function of subclasses? The Ig1, 3 and 4, they can cross the placenta and protect the fetus as we have discussed earlier. IgG3 also activates the complement. Ig1 and 3 binds with the Fc receptor on the phagocytic cell, monocytes and macrophages, and mediate opsonization. Okay, so these are the biological function of subclasses. Now come to clinical highlights, which is very important in regards to IgG, called Rh isoimmunization. We all know what is Rh. So Rh may be positive or negative, like B positive, B negative. So this clinical condition occurs when the pregnant mother is Rh negative and the fetus she carries contains Rh positive blood group. Okay, so during delivery, some fetal blood enters into the maternal circulation, leading to formation of anti-Rh antibodies in the mother. Why? Because mother is Rh negative, so they are not exposed to Rh antibody. When the positive one enters from the fetus to mother, then mother's body treated it as foreign antigen, so the antibody produced against the antigen. Now, in the successive pregnancy, that is in the next pregnancy, if again the fetus being the positive one, then the maternal antibody, which is IgG in nature, they can, being monomer and being small antigen, it can cross the placenta. Now, this antibody which was produced during the first exposure with the fetus antigen, this antibody from maternal blood circulation cross the placenta and enters into the fetus body. Then what happened? Fetus is Rh positive. So, when these antigen exposed with the antibody, they interact and the hemolysis occurs, severe hemolysis, and this leads to hemolytic jaundice or neonatal jaundice, and in severe case neonatal death, even the fetal death can occur or miscarriage can occur, and this whole array of symptoms is called the hydropsis fetalis or hydrops fetalis. It is very serious condition and that is very common also. So in your gynae ward you will face it, and what will be the remedy? The remedy is rather very simple, that is we have to inject anti-Rh antibody within 24 hours of exposure. That is after first delivery, when the mother exposed to the fetus antigen, then within 24 hours of parturition, we should inject the anti-Rh antibody. So this antibody fight against the fetal antigen and destroy it or neutralize it. So the maternal body cannot produce the antibody against the antigen. Okay, so in the successive pregnancy, the fetus will be protected from the hydrolysis or hemolysis. Okay, now goes to immunoglobulin A. This immunoglobulin A, it constitute 10 to 15% of the total immunoglobulin, and it presents in the secretion, body secretion like milk, saliva, tears, respiratory tract secretion, digestive tract, genitourinary tract secretion, etc. So it present in the body secretion or body fluid. In the serum, it exists as monomer, but in the external secretion, it exists as dimer. So this the dimeric form which excreted or secreted it is called secretory immunoglobulin. This secretory immunoglobulin dimer, so how they are connected? They are connected via J chain and via secretory component. This pink one is secretory component and the black one is J chain. So they are connected via J chain and secretory base. The half-life of immunoglobulin A is 6 to 8, whereas in case of IgG it was 23 and it is 11s, whereas in IgG it was 7s. Okay. So What is the how the secretory IgA form? First, from the plasma cell antibody are produced. So IgA produced. The dimeric IgA, when comes to the surface of the epithelial cell, it has got its receptor, which is called poly G receptor. So after binding with the receptor, it endocytosed within the cell and transported towards the lumen. In the luminal side, the enzymatic cleavage occurs. Portion of the poly G receptor remain attached with the IgA and the rest released. So here, which portion of the poly G receptor remain attached with the secretory IgA, which secreted from the luminal side of the epithelium? It is called the secretory piece. So what is the function or role of secretory piece? The secretory component helps protects the IgA from digestive enzymes and denaturation by bacterial protease. Protects the IgA from digestive enzyme and denaturation by bacterial proteases. Now, what are the functions of IgA? It provides local immunity, and one important function of IgA is immune elimination. What is immune elimination? It eliminates the microorganism before invasion. How? Because IgA binds to the surface antigen of the microorganism and prevents its attachment and invasion of mucosal surface of the respiratory and digestive tract. So, it prevents even before the invasion. It eliminates the attachment and invasion. So, it is called immune elimination. The secretory IgA provides important line of defense against some very common organism like Salmonella typhi, responsible for typhoid, Vibrio cholerae, which is responsible for cholera, gonorrhea, influenza virus, etc., polio virus, etc. So we all know the mass immunization of polio virus is due to the secretory property immunity that is provided by the IgA. Okay. So here the herd immunity provides by the IgA. Secretory IgA present in breast milk, which protects the newborn during the first months of life. We all know that colostrum is very important, very healthy for newborn. We should advise for colostrum feeding. That is the first dense, thick, yellowish milk is called colostrum, which is just produced just after production of after delivery, and this colostrum is very much potent source of IgA, okay, and gives protection to newborn against various type of infection. Now it activates complement not by classical pathway, okay, by alternative pathway, promotes phagocytosis and intracellular killing of microorganisms. Now come to immunoglobulin M. This M accounts for 5 to 10% of total serum protein, and it is pentameric. Penta means 5. So they are pentameric, and how they attached? This each monomer attached via J chain, and I am showing you where is the J chain. Here is the J chain, so they are attached via J chain and via disulfide linkage. Here the disulfide bonds between the monomers, so intermonomeric disulfides bonds. So via disulfide bonds and via J chain, these monomers are connected to form the pentameric form of the immunoglobulin M. So they must be heavy. Highest molecular weight of all immunoglobulin is of immunoglobulin M, and this is almost 9 to 10 lakh. This is also called the millionaire molecules. So half-life of the IgM is 5 days, which is very less compared to IgG. Now IgM, 80% of the IgM present intravascularly and present in low concentration in intercellular tissue fluids. Intravascularly they present but low in intercellular tissue fluids. They cannot cross placenta. Obviously, they are very high molecular weight, pentameric in nature, very large molecule, very high molecular weight molecule. So they cannot cross the placenta. But it is produced by the fetus in earliest time of their life. That is, in the 20 weeks, fetus can produce the IgM molecule. Okay. So it is the first immunoglobulin which is produced by the fetus, and also the first immunoglobulin which is produced in primary response. Primary response. What is primary response? Primary response means when we are first encounter a pathogen or antigen. The antibody produced in the first response, it is called the primary response. Okay. So IgM is the primary antibody of primary response, whereas IgG is the antibody of secondary response. Now, it is relatively short-lived, hence it's demonstrated in the serum, indicates recent infection. So, first of all, it produced against primary response as primary response, and it also indicates recent infection because half-life is very small compared to IgG, where it was 23 days. Okay. Monomeric IgM appears on the surface of the unstimulated B lymphocytes, acts as receptor for the antigen. Okay. IgA eliminates the entrance, and whereas IgM appears on the unstimulated B cells, acts as receptor for the antigen. So it acts as primary defense. Okay. Now, what is the function? We already comes to know that it is secreted during the primary exposure and marker of recent infection. It activates complement via classical pathway just like IgG, and it's believed to be responsible for protection against blood invasion by microorganisms. It agglutinates bacteria and cause opsonization and immune hemolysis. So, it has got several utility. As it is the primary antigen which antibody which produced against the it is the first exposure. So if it is present in newborn, it means infection. So in newborn can produce the antibody from 20 weeks. So if it is present in newborn, it indicates infection, and it indicates recent infection also. Okay. So clinical highlights. Other than primary response, it is also called the natural antibody. Primary antibody and as well as natural antibody. Why it is called natural antibody? Natural means which antibody is present within us without any exposure. Suppose think you have the blood group A. Okay, that means you have A antigen. So your antibody, blood group antibody present within you is anti-B antibody. Whereas the person who have B group antigen, they have anti-A antibody. So that during any kind of mismatch encounter, they agglutinate or destroy that particular mismatched cell. Okay. So this presence of anti-B antibody in case of A blood group person or anti-A antibody in case of B blood group person, they present naturally without any previous exposure. So this is called natural antibody, and these antibodies are IgM type. IgM cannot cross placenta. Remember that it cannot cross placenta. So if there is any incompatibility, generally no symptoms produce. Maternal and fetal incompatibility will not produce any symptoms like IgG, where the severe symptom is called hydrops fetalis. Now come to immunoglobulin D and E one by one. Both the immunoglobulin D and E were present in very minor quantity in the serum. Immunoglobulin D 30 microgram per ml and immunoglobulin E 0.3 microgram per ml. They also constitute very feeble amount of total immunoglobulin, 0.2% of total immunoglobulin in the case of IgD. They both has got very similar structure with IgG, and they both exist as monomer. IgD and immunoglobulin D and E both exist as monomer and very similar structure with the IgG molecule. Okay. For IgD, their role in the serum is very uncertain role, so not very well defined. But it has been observed that IgD present together with the IgM in the unstimulated membrane of the unstimulated B lymphocyte. So IgD together with the IgM is major membrane bound immunoglobulin on unstimulated B lymphocyte, even if its role in the serum is uncertain. Okay, it acts as recognition receptor for antigen, but it cannot bind complements or activate complement. So IgD. Now comes to immunoglobulin E. Already have told you that it is also present in very minor quantity and comprise very small portion of the immunoglobulin structure like immunoglobulin G, and also present as monomer. But other important thing for IgE is it is heat labile. That means it can be destroyed at high temperature at 56 degree in 1 hour. The IgE can be destroyed. One peculiarity of IgE is that it has got 4 constant region in the heavy chain CH1, 2, 3, 4. So there are 4 constant region in the heavy chain. It is present only in the immunoglobulin E. It also cannot cross the placenta because it's also very high molecular weight. Weight is 1,90,000. Now IgE is very important for clinical purpose, not like IgD. It is very important for clinical purpose. So why? Because Ig is the main antibody for allergic response or type 1 hypersensitivity response, like hay fever, asthma, anaphylactic shock. So it may be sometimes life-threatening too. Okay, it gives protection, and it is the main antibody to give protection against helminthic infection. So how Ig is the most, Ig is the primary or preliminary antibody for the allergic response. See, Ig binds with the Fc receptor on the membrane of the basophil and mast cell. Basophil and mast cells are the cells which, if they degranulate, they produce the allergic components like histamines and other components which produce allergy. Okay, so basophil and mast cell, they are the storehouse of these allergens. Okay, so if an allergen comes and they cross binds with the 2 different Ig molecule, the allergen binds or stimulate the 2 Ig molecule by binding with the both. Okay, so when 2 Ig molecules on the surface of these cells means that is the blood basophil and the mast cells, these cells are cross-linked by binding to the same antigen, then the cell, this mast cell, degranulates. So degranulation of the mast cell or blood basophil occurs, which release the substance that produce allergy like histamine. Okay, and this is the pharmacological mediators of allergic reaction, even the anaphylaxis. Anaphylaxis means severe type of hypersensitivity reaction against any allergen or antigen. Okay, so severe inflammatory response, that peak occur within 30 minutes, and it sometimes, if the proper protection will not give, proper remedies will not provide, so it may be life threatening also. Okay, now here is the comparison between the several classes of immunoglobulin, that is IgG1, 2, 3, 4, IgM. A also have 2 types, A1 and A2, G, D and E. You will go through it after the class. Now, we will discuss in brief the major function of IgG, just the highlight. You also know, in the meantime, you will come to know that IgG What is that? IgG, antibody of secondary response. Okay. Primary response antibody is IgM. IgG, low molecular weight, so it can cross placenta, produce hydrops fetalis and activates complement by classical pathway. Okay. IgA, main antibody for secretion, secretory IgA. It cannot fix complement by classical pathway but can fix complement by alternate pathway. IgM, antibody of primary response, it can fix complement by classical pathway but cannot cross placenta because it is high molecular weight and pentamer. IgD, it present with IgM on the surface of the unstimulated B cell where it acts as receptor for antigen. IgE, it is the mediators of immediate hypersensitivity reaction or allergy by causing release of mediators from the mast cell and basophil upon exposure to antigen or allergen. So, it is the main mediator of hay fever, asthma, other allergic reaction, anaphylaxis shock, etc. It cannot fix complement and it is the main host defense against helminthic infection. Now, after knowing the structure and the function of individual immunoglobulin, let us discuss about the function of immunoglobulin as a whole. So, first function is antigen binding obviously, and others are called effector function which comprise complement binding, placenta transfer and binding to the Fc receptor. Now, first antigen binding. So, main function of antibody obviously binds with the antigen. So, where is the antigen binding sites? We all know that is a variable region of both light and heavy chain present near the N-terminal end, and in these variable region which comprise the N-terminal end variable region of both the light and heavy chain, it comprises the antigen binding sites. So, in single immunoglobulin molecule there are two such region, number 1 and number 2. So, any antibody molecule or immunoglobulin molecule can bind with two antigenic determinants. So, valency, minimum valency for antibody or immunoglobulins are 2. Now, what is valency? Valency means the number of antigenic determinants that an individual antibody molecule can bind. So, it is minimum 2 for any antibody and it can be more. Just like in case of IgA, dimeric IgA, secretory types, here each monomer binds with two antibody. So, valency as a whole is R4. But in case of IgM, pentameric structure, so it can bind 10 antibody at a time. So, valency 10. Another two terms are very important in this regard. One is affinity, another is avidity. So, what is affinity? Affinity is the sum of non-covalent forces between antigen binding sites of the immunoglobulin and the antigen itself. That means it is the attraction or binding ability of antibody with its antigen via single epitope or single binding sites. And what is avidity? Avidity describes the overall binding affinity of antibodies to the antigen. That means the sum of affinity. Affinity means binding ability with the single epitope, and avidity is the quantitative term or semi-quantitative term. That means overall binding capacity of antibodies with the antigen. Okay. So, affinity and avidity. Now, the effector function. So, what are the effector functions? Effector function number 1 is complement fixation. Number 2, binding to various cell types. That is, the immunoglobulin molecules can bind with various cells like phagocytic cell, lymphocyte, platelet, mast cell, basophils, various cells with its Fc receptor. Which of our cells has got the complementary receptor for binding with the immunoglobulin? Immunoglobulin can bind with these cells via Fc receptors. And after binding, the cell perform their action, that is opsonization and neutralization of the toxin. Another effector function is placental transfer of immunoglobulin. Now, one very important topic in this regard is called ADCC, that is antibody dependent cell mediated cytolysis. Antibody dependent cell mediated cytolysis, ADCC. What is this? IgG molecules can bind with microbes. Here's the microbes, antigen bacterium covered with IgG antibody. Okay. Now, as various cell has got the receptor for this antibody like macrophages, this Fc portion, via Fc portion, this antibody can fixed with the or attached with the macrophage. Fc portion present in the stalk. So here in the receptor present on the macrophages binds with the Fc portion of the IgG antibody, which binds with the microbes or bacterium. Okay. So after binding, what occurs? That is phagocytosis or internalization. Internalization is also called the opsonization. So phagocytosis or internalization of the bacterium is called the opsonization. Okay. So opsonization occurs. After opsonization, the antibodies coating the target cell sensitize the cell, particular cell which opsonize the bacterium covered with antibody like macrophages to kill the cell or lyse the cell. That is why it is called antibody dependent cell mediated cytolysis. That is, antibodies helps the target cell or sensitize the target cells for killing the antigen by macrophages. This is called antibody dependent cell mediated cytolysis. Only IgG has got this property. Okay, remember that only IgG can act as this type. Okay. Now, the next function, that is placental transfer. It has been seen that some immunoglobulin receptor also present in the placental trophoblast. Thus, like there is a receptor present in various cell types, some receptor also present in the trophoblastic cell of the placenta. And this type of receptor helps in transfer of the immunoglobulin across the placenta to provide the immunity to the fetus. Okay. Now, as this result, the maternal antibody can protect the fetus and newborn. Now, after discussing the structure and function individually and as a whole, let us discuss about the clinical significance of this immunoglobulin. So, there may be agammaglobulinemia, that means no production or absence, complete absence of globulin. This is gamma globulin, that is agammaglobulinemia. Number 2, hypogammaglobulinemia, that means produced in lesser quantity than the normal, so hypogammaglobulinemia. Number 3, hypergammaglobulinemia, or produced more than the normal. Okay, so let us discuss few important disease one by one. So there are paraproteinemias or monoclonal immunoglobulin. These are few importance are multiple myeloma, Bence-jones proteinuria, heavy chain disease, Waldenstrom macroglobulinemia, amyloidosis, hypergammaglobulinemia as a whole, and hypogammaglobulinemia as a whole. So first one is multiple myeloma, which is very important. Multiple myeloma is the malignancy of single clone of plasma cell. We know that globulin or immunoglobulin produced from the plasma cell. So it is also called the plasmacytoma. Oma means tumor. So it is the tumor of plasma cell, single clone of plasma cell in the bone marrow, and it constitutes almost 1% of all the cancer and common in case of females in the age group of 45 to 60 years. Okay. Most of the cases, the type of clone of plasma cell which produce IgG are affected. That means most of the time, almost 75% of the times, it is IgG type. In some cases, that is 25% cases, in case it is IgA or IgM type. Okay. There is a type IgD type which present in younger adults, not in the 45 to 60 years age group, younger adult, but it is more virulent, though less common, but it is more virulent. Now, what are the typical clinical features? You will study multiple myeloma in detail in later professional exam, but just a brief. Okay. So, carcinoma means cancer, and cancer means crab, and the symptom also has got mnemonic crab. C for calcium is increased, that means hypercalcemia. R for renal failure, A for anemia, B for bone lesion, lytic bone lesion. So why calcium increase? Because of the lytic bone lesion. That means from the bone osteolytic lesion, from the bones, the calcium released into the circulation. So there is hypercalcemia. There is severe production and destruction of the particular immunoglobulin. So the protein increased, and also the calcium, kidney can't handle the stress. So renal failure, and the bone marrow cannot produce the hemoglobin properly. So anemia. Now, due to this lytic bone lesion, spontaneous pathological fracture is very common in the weight bearing joint, because bones get full of osteolytic lesion, not solid enough, no calcium deposited. So spontaneous pathological fracture, reef fracture, vertebra fracture may occur, and these cause very severe pain also. And paraproteinemia, that means monoclonal gammopathy. A particular clone of immunoglobulin increase leads to proteinuria, and there is raised beta macroglobulin. Bone marrow examination obviously shows the malignant plasma cell, and X-ray shows the osteolytic lesion which is punched out. That means someone punched the bones. Seems that someone punched the bone, the bones get broke. So whole type of, that is punched out osteolytic lesion, is very typical feature of this multiple myeloma. Now, our concern is electrophoretic pattern, which is very much diagnostic of multiple myeloma. So, in case of multiple myeloma, the characteristic pattern is the presence of sharp and distinct band called M band with narrow spike. This M doesn't mean the IgM. Remember that. Don't take it as IgM. This M means monoclonal band or multiple myeloma band. Okay. So there is sharp distinct band called M band with narrow spike, which position between beta and gamma portion of the globulins in the electrophoresis. Okay. These M bands almost replace the gamma globulin band due to the diminished. Obviously, if abnormal type increase, normal type diminish. So it almost replaced the normal gamma globulin band due to the diminished synthesis of normal gamma globulin. So let us see the pattern. This is the normal electrophoretic pattern of normal serum. Okay, that means albumin is most abundant, followed by alpha 1, alpha 2, beta 1, beta 2. They merge, then followed by gamma. But in case of multiple myeloma, all others decrease slightly, and a sharp spike of M band, that is monoclonal band, almost replace the gamma region. Okay, so this is the typical electrophoretic pattern for multiple myeloma. Now comes to the Bence-jones proteinuria. It is also very much interesting. Bence-jones proteinuria. Here, why it is called Bence-jones? Because Henry Bence-jones first described the disease. So, according to his name, it is called Bence-jones proteinuria. In this case, Bence-jones protein, a particular type of protein excreted in the urine, which is often damage the renal tubule, and excess synthesis of light chain occur. This Bence-jones protein composed of the light chain, excess synthesis of light chain, that means either kappa or lambda varieties of light chain produced in excess quantity, and these type of light chain protein, which is called Bence-jones protein according to its inventor's name, it excreted in the urine, okay, and caused the renal damage. This Bence-jones proteinuria also present with 20% patients with multiple myeloma. Okay. Now, what are the specific tests for Bence-jones proteinuria? One is very simple test called the classical heat test, which is not diagnostic obviously, but it can at least suspect the Bence-jones proteinuria. In this test, what happens? That is, it's BJP or Bence-jones protein, not Bharatiya Janata Party. It has got special property of precipitation when urine is heated to 40 to 50 degrees centigrade. Okay, so in the 40 to 50, you take the urine sample of the patient, you heat to 40 to 50 degree in the water bath or in the incubator, then the proteins precipitate, and if protein precipitate, what appear? White clump, isn't it? You know that. So if we further increase the heat up to 80 degrees centigrade, then again that precipitation get dissolved. Okay, so the urine become clear, and if we cool the sample again, the precipitation reappear in the range of 40 to 50 degrees centigrade. So first appearance of the coagulum in the 40 to 50 degrees centigrade, then dissolve after increasing the temperature to 80 degree, again reappear when we cool down the sample to 40 to 50 degree. This is called the classical heat test. Next test is obviously electrophoresis, which is very important for any type of monoclonal gammopathy. And another confirmatory test is called Bradshaw test. Bradshaw test. Here, white ring of precipitate will occur if the urine is layered on few ml of concentrated HCl or HNO3 also. So we take few ml of concentrated HCl, then we add the patient sample or patient urine drop by drop, and the white ring of precipitate appear at the junction. It is called the Bradshaw test for Bence-jones proteinuria. Remember that Bence-jones protein is light chain protein, low molecular weight protein containing either lambda or kappa chain. Next one is Waldenstrom's macroglobulinemia. So in this case, the IgM type of immunoglobulin clone gets proliferated. So malignant proliferation occurs in IgM clones. In case of multiple myeloma, it is mainly IgG. And here males are most commonly affected, rather female. Since we all know that IgM is very heavy and pentameric molecules very heavy, so in this case aggregates of precipitation forms, and this increase the viscosity of the serum. So highly viscous serum form. How it is diagnosed? It is diagnosed by CR test. CR test is what? We take two test tube. One is for control and another is for patient. Okay. We take water in both the test tube. Then apply water or normal serum in the one test tube and patient serum in another test tube. If the patient is positive for Waldenström macroglobulinemia, then white precipitate, globular precipitate forms in the test tube for the patient, whereas in case of control, the normal serum sprayed within the water. Normal serum just sprayed within the water, and the patient serum forms the due to the hyperviscosity, it forms globular precipitate. Okay, so this is called CR test for Waldenström macroglobulinemia. Now, heavy chain disease. In case of heavy chain disease, what happened? That is, some portion of H chain gets deleted during synthesis and cannot join with the light chain to form the normal immunoglobulin. Rather, it excreted through the urine, and it may be gamma chain or maybe alpha chain. In case of gamma chain, it causes hepatosplenomegaly, lymphadenopathy, etc. In case of alpha chain disease, it causes abdominal lymphoma, malabsorption syndrome. Okay, so light chain disease is called Bence-jones proteinuria, which is important one, and heavy chain disease mainly composed of gamma chain disease or alpha chain disease. Okay. Now amyloidosis. Amyloidosis is also the disease of light chain. Here 20% patients of myeloma develop amyloidosis. How? Because the light chain fragments of the clone which proliferates, light chain fragments of that clone of plasma cell deposited within the tissue, in the liver, kidney, intestine, even within the brain. Okay, so they develop typical symptom due to the deposition of the light chain fragments within the tissue. Okay, this is called amyloidosis. Now, brief summary. First, you have to remember the definition, the structure, very important. You have to practice the drawing. Okay, you have to draw the structure. Whenever it comes, you have to draw. Okay, Y-shaped, tetrameric, heterodimeric, heavy chain, light chain, variable region, constant region, hinge region, then that is disulfide linkage, and the various portion, that means FAB portion, FC portion, and don't forget to mention the carbohydrate attached with it. Okay, then the individual immunoglobulin, they have got some specialty, that is the 5 immunoglobulins based on their heavy chain present in them, and they are differ from the Weidbach coefficient, that means S, and valency also differ, and their main function also differ one from another. Like IgG is the antibody which can cross the placenta from maternal to fetal circulation and can produce hydrops fetalis. Okay, IgG is the antibody which produced in the secondary response. IgM, antibody of primary response. So it denotes recent infection, and the first antibody produced in the fetus itself. Okay, IgD and IgM present on the unstimulated B lymphocyte. IgE is the main antibody related with the allergic reaction and type 1 hypersensitivity reaction, also provides protection against helminthic infection. Okay, so hope you will understand and you will go through the slides and your book. Okay, so student, please stay home. Hope you will stay safe in your home, and thank you very much.